Welcome back to this next video in which we are studying headaches. Okay, so we're currently in the process of discussing the pathophysiology of migraine headaches, and what we have seen so far is that the phenomenon of cortical spreading depression is responsible for both migraine headaches with aura and migraine headaches without aura. Remember, when you're suffering from a migraine headache that didn't have a preceding aura, we think that the reason for that is that the cortical spreading depression has occurred in some portion of cerebral cortex that doesn't give rise to an aura when the cortical spreading depression occurs there, but which will nevertheless activate the overlying meningeal nociceptors. Now, we will discuss how cortical spreading depression is going to activate meningeal nociceptors in much more detail in the next video. At the moment, what I want to continue on with in this video is discussing the genes responsible for familial hemiplegic migraine of the different types. And remember, the reason we're doing this is because these are monogenic cases of migraine, and they're monogenic cases of migraine that give rise to susceptibility to cortical spreading depression, specifically in the primary motor cortex, which is an area of cerebral cortex that normally in the polygenic cases of migraine, you don't normally have susceptibility to cortical spreading depression there, and therefore usually you don't get a motor aura. The areas that are commonly affected if you are going to get an aura is the occipital pole, giving rise to the visual aura and potentially the sensory aura. Okay, and the reason we're going over these is because these are monogenic and they might give us hints, and indeed they are giving us hints, about what it actually is that gives you susceptibility to cortical spreading depression. So we've so far done familial hemiplegic migraine type 1, and we've seen that this is caused by having a single copy of a disease-causing allele of the CAV2.1 alpha-1 subunit of voltage-gated calcium channels. This will, if you have it, give rise to you having some dysfunctional PQ-type voltage-gated calcium channels in your brain, and somehow this puts you at risk of developing cortical spreading depression, specifically of developing cortical spreading depression in the primary motor cortex, which normally in the polygenic cases of migraine does not happen. So this is showing us that electrolytes and the control of the movement of electrolytes between the intracellular and extracellular spaces may have a big role in cortical spreading depression, which isn't surprising because it is, an electrical organ, the brain, so electrophysiology probably plays a role in this process. Okay, so that's the interesting insight then that we got from familial hemiplegic migraine type 1. Let's now talk about familial hemiplegic migraine type 2. After familial hemiplegic migraine type 1, it's the second most common form of familial hemiplegic migraine. Now, this is caused by mutations in a different protein, but another protein that is involved in controlling electrolytes and electrophysiology. It's caused by mutations in one of the subunits of sodium-potassium ATPases, the sodium-potassium pumps. Okay, so these are extremely important objects. These are the objects in cell membranes that move free sodium ions out of the cell and 2 potassium ions into the cell to maintain the sodium gradient across the cell membrane and the potassium gradient across the cell membrane. And to do that, it's an active transport process because they're moving it against electrochemical gradients often, and they will hydrolyze ATP to do that. So let me just draw you a picture then of the structure of sodium-potassium ATPase, and then we'll discuss the specific subunit that has alleles which can give rise to familial hemiplegic migraine type 2 if you have one of them. And again, remember, all of these are autosomal dominant conditions. So, the structure of the sodium-potassium ATPase, let me get another colour. So, it's made out of 2 subunits. The major subunit, which is equivalent to the pore-forming subunit of the voltage-gated calcium channels, is called the alpha subunit. This is the one that actually does the moving of the sodium ions and the potassium ions and the hydrolysis of ATP. But then you also have an accessory subunit stuck onto the alpha subunit, like so, which is called a beta subunit. So, sodium-potassium ATPases, they consist of 2 subunits, an alpha subunit and a beta subunit. And as I say, the alpha subunit's the main one. That's the one that actually moves let's draw it on here. It's going to move, complete up that arrow, 3 sodiums out of the cell membrane, so orienting you, this is the inside and this is the outside, and in exchange for that it will be moving 2 potassium ions into the cytoplasm of the cell, and this maintains the sodium and potassium concentration gradients across the cell membrane. Remember, sodium is very high outside of the cell and it's very low inside of the cell. I think it's 145 millimolar outside of the cell, whilst it's 12 millimolar inside of the cell. So let me put some of these fancy statistics that I'm showing off with actually on the picture. So 145 millimolar is the concentration of sodium outside the cell, and inside the cell I think it's around 12 millimolar, so that's around a 12-fold gradient. And there's an even bigger concentration gradient of potassium. Outside the cell, potassium's concentration is around 4 millimolar, whereas inside the cell it's around 155 millimolar. So these concentration gradients of sodium and potassium across the cell membrane, they have to be maintained by something, and that's maintained by the sodium-potassium ATPase, because sodium is continuously moving back into the cell through the leaky sodium channels, and potassium is continuously moving out of the cell through the leaky potassium channels. So if you're going to maintain these concentration gradients, you need to have something reversing that movement, and indeed those are the sodium-potassium ATPases. Now, this requires energy to actually operate, so this thing will be hydrolyzing ATP to ADP and inorganic phosphate. So ATP will be being hydrolyzed to ADP and inorganic phosphate. So again, the beta subunit is a modulatory subunit. It changes properties of the alpha subunit. So for instance, the rate at which it's doing this, its position in the cell, it monitors things like that, like the auxiliary subunits of the voltage-gated calcium channels alpha 1 subunit did. Okay, so again, in the beautiful, simple world, it would be the case that there was just one gene for the alpha subunit of sodium-potassium ATPases and one gene for the beta subunit of sodium-potassium ATPases. Unfortunately, the world is much more complicated than that, and there are 4 separate genes for alpha subunits and 4 separate genes for beta subunits of sodium-potassium ATPases. And again, I'm not counting there the 2 copies on the homologous chromosomes. So if you counted the 2 copies on the homologous chromosomes, it would be 8 different alpha subunit genes and 8 different beta subunit genes. But we have overall 4 different types of alpha subunit gene and 4 different types of beta subunit gene. Okay. So what are the names for those? So they're given very simple names, thankfully. So everything begins with ATP, because we're talking about an ATPase that breaks down ATP. Fundamentally, it is an enzyme that breaks down ATP. And because it's such an important ATPase, we call it the ATPase 1, so we put 1 there. And then depending on whether we're talking about the alpha or the beta subunits, we either put A or B next. So you could put A if we're talking about genes for alpha subunits. If we're talking about genes for beta subunits, we put a B, and then they're just called 1 to 4. So there is the ATP1A1, the ATP1A2, the ATP1A3, and the ATP1A4, which are the 4 different genes for alpha subunits of sodium-potassium ATPases. There are then the ATP1B, 1 to 4 as well, which are the 4 different genes for the beta subunits of the sodium-potassium ATPases. Alright. So there's the background about sodium-potassium ATPases. Familial hemiplegic migraine type 2 is going to be caused by having a disease allele in one of these ATP1A genes, and specifically the one that is the culprit is ATP1A2. So the culprit for familial hemiplegic migraine type 2 is ATP1A2. So again, this is a gene. You'll have 2 copies of it. It's on an autosome, so you'll have 2 copies on the 2 homologous chromosomes. And again, just like this picture here, there will be a huge number of different alleles that you can have in those 2 gene sockets in those positions. A bunch of those alleles will then be familial hemiplegic migraine type 2 disease alleles. And if you have just one of those in one of the sockets of ATP1A2 that you have, you will get familial hemiplegic migraine type 2. Okay, and again, there are multiple different disease alleles. There's not just one allele that's in this great big set here that can cause familial hemiplegic migraine type 2. There's loads of disease alleles of the ATP1A2 gene that if you have one of them, hence it's dominant, you will get familial hemiplegic migraine type 2. So what is this showing us? That if you do indeed have one of these disease alleles, what will you produce? You will produce a whole bunch of faulty sodium-potassium ATPases. Now again, it won't be all of your sodium-potassium ATPases that are faulty, but some of them will be faulty now. And again, something is going to go wrong with electrophysiology, electrolyte handling, and the voltage across the cell membrane potentially, and that now is going to put you at risk of developing cortical spreading depression, and specifically somehow in the primary motor cortex again. Okay, right, so again, this is backing up the story that we've had so far, that your risk of developing cortical spreading depression is probably encoded in electrophysiological proteins, how well your proteins that handle the electrophysiology of your neurons are functioning. Okay, so finally, to complete the story, let's look at familial hemiplegic migraine type 3, and this now goes to an even more famous actually no, probably not an even more famous, I mean the sodium-potassium ATPase is probably the most famous, but another very, very famous one, which is now voltage-gated sodium channels. So this one is the rarest type of familial hemiplegic migraine, and actually this one, at present, there is only one disease allele that we currently know about, which separates it from the other two where we know about loads of disease alleles of those genes that can cause the disease. So let me firstly remind you of the structure of voltage-gated sodium channels. So I hope you are fine with my abbreviation here. Voltage-gated, N for sodium, Na, natremium or whatever, and then C for channels. So let me remind you of the structure of voltage-gated sodium channels. So it's very similar to voltage-gated calcium channels, except that it's even simpler. You only have two subunits in voltage-gated sodium channels. So let's do it here in pink. You have the pore-forming unit, again, the most important unit, that is actually the one that will open in response to depolarization of the voltage across the cell membrane and will allow sodium ions to move through a tube down the middle that I've drawn on there and move from the outside of the cell into the inside of the cell down its electrochemical gradient. So this subunit here is the alpha subunit of the voltage-gated sodium channel. Now, it has its auxiliary subunits as well, as always, making the structure a little bit more complicated. So there is just one auxiliary subunit in the case of voltage-gated sodium channels, and that is the beta subunit. So this is the structure of voltage-gated sodium channels. Again, the auxiliary subunit monitors its behavior. It modulates its behavior. It alters the voltage at which it will activate. It alters things like how quickly it will activate. It alters things like the location of the voltage-gated sodium channel in the cell membrane. Things like that. Okay, so again, the same complexity is going to strike here, that there is not just one alpha subunit of voltage-gated sodium channels. And by the way, familial hemiplegic migraine type 3 is going to arise because of a problem with an alpha subunit of voltage-gated sodium channel. So we're not going to discuss the different beta subunits. So there is not just one alpha subunit of voltage-gated sodium channels. There are actually 9 different genes coding for voltage-gated sodium channels, again, not counting the homologous copies on the homologous chromosomes. All of them will have homologous copies, making overall 18. But 9 different types of genes for alpha subunits of voltage-gated sodium channels. What are their names? Their names are NAV, so at least someone, when they came up with these naming systems, was being consistent. Because it's a sodium channel, we put Na, and then we put a subscript V, just like we did for the alpha subunits of voltage-gated calcium channels. And then it's NAV 1.1, all the way up to NAV 1.9. So there's only one family of alpha subunits of voltage-gated calcium channels, of alpha subunits of voltage-gated sodium channels at present. That is the NAV1 family, and they've all been named with the point here. So NAV 1.1, NAV 1.2, NAV 1.3, all the way up to NAV 1.9. Those are the 9 names for the alpha subunits of voltage-gated sodium channels. Okay, so you can use 9 different types of alpha subunits in this position when you're making voltage-gated sodium channels. Again, there are multiple different genes for the beta subunit of voltage-gated sodium channels, but we're not going to go into that. Okay, so which one of these genes for an alpha subunit of voltage-gated sodium channels then is it that is responsible for familial hemiplegic migraine type 3? It is the NAV 1.1 here. This one, which I'm boxing now, is the one that can go wrong in familial hemiplegic migraine type 1. So, this time it's more simple actually than the other two cases, because now if I was to draw this picture again, there is only one disease allele. So again, it's autosomal dominant, so this gene, NAV 1.1 gene, you'll have two copies of it. It's on an autosome. I don't know specifically what chromosome it's on, but it's on one of the autosomes. You'll have two copies of it, so you can choose, you've got two sockets effectively, and you need to put alleles of the gene in those sockets. There will be loads of different alleles of NAV 1.1 in the population, again, a great big set like so, and in your sockets you're going to have two of these alleles. They could be the same allele in both sockets, but they're unlikely to be. Now, in this great population of alleles of NAV 1.1, at present, there is only one known allele that, if you have that present, and you only need one of them because it's dominant, remember, will give you familial hemiplegic migraine type 3. That will probably change in the future. We will probably find more alleles, disease-causing alleles of NAV 1.1, but at present we only know about a single disease-causing allele of NAV 1.1, and you only need one copy again of that disease-causing allele to actually get the disease familial hemiplegic migraine type 3. So, how is this going to work then? If you have that disease-causing allele, you will make the protein, you will make this faulty alpha subunit here. Therefore, some of your voltage-gated sodium channels will have this faulty alpha subunit in, and again, this is going to distort electrophysiology somehow, and that seems as though it's going to give you that susceptibility to developing cortical spreading depression. And again, somehow this specific protein seems to be important in the primary motor cortex, so again it seems that you have this susceptibility to developing cortical spreading depression in the primary motor cortex, giving you familial hemiplegic migraine. So those are the three different forms of familial hemiplegic migraine and the genes responsible for them. And they have given us this tantalizing clue as to what it is potentially that is important when we're assessing the risk of cortical spreading depression in someone's brain. Okay, right, so we will have a break here. I only wanted to finish off my discussion of cortical spreading depression in this video. I don't want to go yet onto the discussion of the activation of the meningeal nociceptors. We'll do that now in the next video. So come back, join me for the next video, and we'll complete the story of the pathophysiology of migraine by discussing how this phenomenon of cortical spreading depression occurring in a region of cerebral cortex is going to activate the overlying meningeal nociceptors.